Welcome to Measuring Carbon in Vegetation. This video demonstrates how to measure the carbon found in vegetation, or non-treed plant material. It introduces some standardized field methods for determining the carbon stock of non-treed vegetation of different ecosystems throughout Canada, including shrublands, grasslands, tundra, and wetlands. You may also choose to combine these methods with carbon measuring in trees to estimate the broader carbon stock within a forest ecosystem. The carbon stock of non-treed vegetation in Canada is much smaller than it is for trees, about 0.2 billion tons. Still, this vegetation contributes significantly to long-term carbon storage by altering its environment and producing materials that resist decomposition. In this video, we will introduce a set of standardized field methods for determining the carbon stock of non-treed vegetation of different ecosystems throughout Canada. Before sampling any vegetation, it is important to think about site selection. For example, where the plots are to be set up, how many are needed, and what materials are required. Your study area encompasses the entire area that you wish to investigate. Within this large area, specific smaller study sites are identified to capture the variation of the area. Sites can be distinguished by changes in vegetation cover, elevation, ecosystem type, or other geographical features that vary across the study area. Within each site, 1 to 5 plots can be set up, depending on the nature of the site. The more natural variation that exists across a site, the more plots that are required. For further details on establishing sites and plots, please see the accompanying guide. Once you have defined the study area, sites, and plot locations, you can begin to gather vegetation data. Here is a list of the required equipment. 2 plot types are required to sample all non-treed vegetation within the study area. The first are medium vegetation, or shrub plots, which are typically between 16 and 100 meters square. All vegetation between 0.5 and 2 meters in height will be sampled in this plot. The second is small vegetation, or microplots. These plots are 1 by 1 meter in size. All vegetation below 0.5 meters in height will be sampled in these plots. Plot locations should be mapped out ahead of time based on a standardized sampling design. For detailed instructions on how to set up a study site, please refer to the accompanying guide. For all plots, measure GPS coordinates, including longitude, latitude, and elevation of the plot center. Record these measurements in your notebook. Next, you'll need to map out the border of your plots using a measuring tape. If a compass is available, align the width of the plot along the east-west axis and align the length of the plot along the north-south axis. With the plots mapped out, you can now conduct surveys. For the medium vegetation plots, the goal is to systematically identify each individual plant species and obtain measurements for plant volume. You will then use this information to estimate total biomass. Species identification can be done using a dichotomous key or via a mobile species identification application such as Google Lens, LeafSnap, and Seek by iNaturalist. It also helps to familiarize yourself with the local plant communities before doing surveys, as this step can sometimes be tedious. There are several online resources available to find this information specific to your area. If the species in question is a small tree, measure the diameter of the tree stem in centimeters at 0.3 meters above ground level. Record this measurement in your notebook. If the species in question does not have a single woody stem, but rather has multiple branches emerging together from the earth, then it is a shrub. In this case, measure the plant's volume instead of its diameter. Measuring plant volume requires knowing the maximum height, length, and width of the plant. These measurements are best conducted with 3 team members. Have 2 team members hold the measuring tape on either side of the plant, while a third team member records the measurements in the notebook. Converting these measurements to biomass and carbon stock for a plant requires using a series of mathematical equations, which are provided in the accompanying guide. For estimating the carbon stock for vegetation below 0.5 meters in height, the most common and accurate method is called the clip and weigh method. The first step for all microplot sites is to take photos of the plot directly above the plot center. Make sure the boundary of the plot is visible in the photos. Using a 1 by 1 meter frame can help with this. Section off a quarter of the plot using a square or circular frame with a 0.25 meter square area. Collect samples for all plants in the frame by taking a clipping of every single plant at 3 centimeters above ground level. Put these samples into bags labeled with the species, date, and site identifier or coordinates before transferring to a lab for analysis. For other sampling methods for small vegetation, consult the accompanying guide. Each sampling method detailed in the guide results in an estimate of the total carbon stock of the vegetation in the plots sampled. To calculate the average carbon stock of a medium or small plot, add the total carbon stock of all individual plants measured in each plot and divide this value by the plot size to get the amount of carbon per unit area of the plot. Complete this calculation for all the plots in the site. Then, for each site, add all the plot's average carbon stocks and divide this value by the number of plots in the site. Remember that all the plots are the same size. This is the average carbon stock across the site. Multiply the average carbon stock of the site by the size of the site in meters square to obtain the total carbon stock of each site. But what we're really interested in is the carbon estimate for the entire study area. So, next add up the total carbon stocks for all the sites and divide by the sum of the site sizes. This gives the average carbon stock of the study area. Finally, if you're interested in total carbon stock of the study area, multiply the average carbon stock of the study area by the size of the study area. Congratulations! You now know how to measure carbon in vegetation. In this video, you reviewed the steps for selecting a study area, site, and plots, preparing your plot for measuring, identifying plant species in your plot, collecting data for each plant in your plot, using plant data to estimate biomass, and using plant data to estimate total carbon stocks for a study area. For more information, please consult the carbon measuring guidelines for vegetation and refer back to this video anytime. Happy measuring!